Can I start? Namaskaram. Welcome to KNA. My name is ASK Ashok Kumar. Now we are going to speak about top 30 questions. The best 30 questions extracted from 3 chapters: body fluids and circulation, breathing and exchange of gases, and excretion. So from these 3 chapters, we are going to around 33 questions we planned we are going to make. So along with the questions, okay, along with the question and answers, you need to understand the whole concept around it. I will cover, okay na? So get ready. So everyone, check the questions properly. Or if you, I made like a different way, okay. Going to start the first question I am going to place in front of you. This question is important. First of all, this question is important for the NEET exam, okay. So this question, when you see itself, this is related to the composition of the plasma proteins. First we will make it to the what it called as a concept I will tell you, okay na? Make it to clear. So here I am going to speak about the plasma, blood plasma composition. As we know, this plasma is occupying the 55% of the blood composition, okay. In this plasma consists of, what are the components in the plasma? Okay, already I said this question is important. 1 year given in the NEET exam. Okay, na? This question itself, it is an important one. The plasma composition, first one is a water. 90 to 92% of the plasma is occupied by the just water. That is followed by the proteins. So this protein is occupying the 6 to 8%. The remaining, other than the proteins and water, the remaining is occupying by the other solutes, is occupied by the other solutes. What are these other solutes? Include these other solutes. The pen is not working, sir. These other solutes include nutrients. Next one is a gases. When I am telling itself, you can understand, okay? Nutrients, gases. The next one is a hormones. The next one is enzymes. Next one is excretory substances. So this is the overall composition of the plasma. One more time you can check. Then I will go for the explanation in this one. Okay. This include a plasma composition. Out of the blood, plasma is occupying the 55%. Within this plasma, water is a 90 to 92%, proteins 6 to 8%. Remaining portion is occupied by the other solutes. That other solutes include the nutrients. As we know, the nutrients include a glucose, amino acids, lipids, etc. Next one is a gases. I am speaking in the plasma, gases. Gases means oxygen and carbon dioxide. So just I am asking, so in this gases concept, how much percentage of oxygen is dissolved in the plasma? 97% of the oxygen transported by the RBC, that is a hemoglobin. 3% of oxygen present in the dissolved state in the plasma, if you remember. In case of the carbon dioxide, okay, carbon dioxide 70%, majority or 70% of the carbon dioxide is transported by in the form of bicarbonate. Okay, 20 to 25% up to there, it is transported by the hemoglobin. Next 7% of the carbon dioxide present in the dissolved state in the plasma. You understand? 3% of the oxygen, 7% of the carbon dioxide present in the dissolved state in the plasma. So nutrients I said, gases I said, hormones. Everybody will neglect the hormones, but hormones are very important. Okay, because all the hormones are produced by the endocrine glands. These endocrine glands directly secreting the hormones into the body fluids. Main body fluid is the blood. The next one is a enzyme. Different types of enzymes are present. If you remember, carbonic anhydrase. Large amount of carbonic anhydrase enzyme present within the RBC, but minute quantity also present in the plasma. The next one is a excretory substances. Excretory substance include mainly urea. Urea is there, creatine is there, bilirubin, biliverdin. but mainly is a urea. This is the overall composition of the plasma. I hope you are clear. Okay. Now this question is with respect to all the proteins. With respect to all the proteins, mainly three types of proteins: albumins, globulins, and fibrinogens. Correct na? Albumin main function is what it called as a osmotic balance. Very, very important function. What is the most abundant protein present in the blood? That is the albumin. Albumin responsible for the osmotic balance, osmotic pressure or osmotic balance. Globulins, if you remember, this word is a globulins. Globulins, that is belongs to the antibodies. Antibodies are belongs to immunoglobulins. This function is a defense mechanism. Okay. Then the next one is a fibrinogen. Okay, na? That is involved in the blood coagulation. So first question itself is very important for the NEET exam. I am going forward to the next question. Okay. So in this one, I covered the composition of the plasma. Okay, along with the question. So albumin is responsible for the osmotic balance. So A is belongs to 2. Next one, globulin responsible for the defense mechanism 1. Fibrinogen for the blood coagulation. I am going for option. So 2, I am going for the option 2. Now I am going to the next question. The following components in the plasma are always in the transit in the body. So already I made the composition of the plasma direct with the same explanation only. The following components in the plasma always in the transit. Transit is related to the always in the circular, always in the transport in the this one. That is answer is a nutrients. Direct I am going for the answer. That is answer is a 2, nutrients. Okay. Nutrients like I am telling the NCERT statement. Nutrients like glucose, amino acids, lipid, etc. are always transit in the circulatory system. Okay, that's the direct question. Now, the next one I am going to make the third question. Now, it is your turn. What are the following proteins present in the blood except? The given options you can see, you can observe. The following proteins are present in the blood except. This is a connecting point. This is a connection question between the our what it called as a structural organization. There is a chapter concept is called as a tissues are there na? And body fluids and circulation. So one time you can check and you can tell. So here this one. So among the following protein in the blood except, as we know, one statement I will tell you, we can recollect from the structural organization in the tissue. Among all connective tissues except blood containing the cells that produces the collagen and elastin. One more time I am telling, in all connective tissues except blood, nicely frame the statement. All the connective tissues except blood containing cells that secrete collagen protein and elastin protein. That is except blood. So blood containing, directly you can tell one way. Blood containing albumin protein is there, albumin, globulins, fibrinogen, but not collagen. That is why I am going for the option 2, collagen. Okay. So this is the answer. Comes to the take the collagen. When we heard about the collagen, what called as a intercellular? It is a intercellular, or it is a protein present in the intercellular component or the ground tissue in case of the connective tissue. Okay. So you remember, collagen is the most abundant protein in animals. Don't call it as a mammals. It is the most abundant protein in the animals. The next one is that triple helical structure of collagen. Triple helical structure of collagen was discovered by G.N. Ramachandran sir. G.N. Ramachandran sir. So these 3 points, first I am telling, first statement, question itself is a connection between the two chapters. The following proteins present in the blood except, answer is a collagen. Already I said important statement. Okay. When you are speaking about the collagen, collagen is a very important protein present in the ground tissue of the connective tissue. Second one is that collagen is the most abundant protein in the animals. Third one is a triple helical structure of collagen. There is a photo is there, G. N. Ramachandran sir. Triple helical structure of collagen was discovered by the great G. N. Ramachandran sir. Okay, these 3 points about the collagen. Functions of albumins, globulin, fibrinogen already we covered. Okay, going to the next question. Find the mismatch with respect to of blood cells and with its a percentage. That's very good. Okay, here I am going to cover this directly we can make. So I said now when I started it itself I said along with the question that around the explanation is also required for you. Okay. So with respect to of this one, what is the abundancy order? So pen is stuck in, sir. What is the abundancy order of the cells? If you take the formed elements except platelets, you can put one side. What is the abundancy order means? First one is a RBC, correct na? Most abundant blood cell is a RB. First we can frame the abundancy order. This is a most abundant. This one is a most abundant. The next one is after RBC. Thank you. So most abundant is RBC, that is followed by the neutrophils. So RBC, then after RBC we will go for the WBC. WBC divided into granulocytes and agranulocytes. Here the point is related to the, within this one is a neutrophils, correct na? Neutrophils occupying the 60, thank you sir. Neutrophils occupying the 60 to 65%, okay na? That we can discuss, okay? The next one is called as the lymphocytes. Lymphocytes occupying the 20 to 25%. Next one is a eosinophils. Eosinophils occupying the 2, what it calls here, 2 to 3%. The next one is, before these eosinophils, we need to mention the monocytes also. Monocytes also, correct na? The next one is going for the basophils. The next one is a basophils. Basophils is 0.1, 0.5 to 1%. This is a abundancy order. One time can you check your with the existing knowledge? What is the abundancy order of the cells, blood cells? One is the most, which is the most abundant blood cell, RBC. For example, with this order, okay, RBC, neutrophils, followed by the lymphocytes, followed by the monocytes, eosinophils, followed by the basophils. Okay, these are the things. Which is among the most abundant the blood cell, going for the RBC. Which is most abundant WBC, most abundant leukocyte, that is a neutrophil. Which is the least abundant WBC, going for the basophils. Most abundant WBC is a neutrophil, least abundant WBC belongs to the basophils. Okay, na? That's one. This is a abundancy order. Very nice. Next slides we will cover the functions. Okay. Now you can go for the neutrophil 60 to 65%. That's correct. Eosinophils okay. Basophils okay. Lymphocytes you can see. Lymphocytes is 20 to 25%. Na? This is a incorrect. Mismatch pair. Fourth one I am going to take the mismatch. This is a 1,50,000 to 3,50,000, that is related to the platelets. It's related to the platelets. Okay. So now you can got the composition of the plasma you got, functions of the proteins you got. Next one is the abundancy order of the formed elements also you got. Okay. Next one. Among the fraction of the formed element, now this is a time for you. Okay, now one time you can check the question. Among the following fraction of formed element is not a cell. What about your opinion in this question? Among the following fraction of the formed element, which is RBC, Option 2, lymphocytes. Next one is a platelets. Next one is a neutrophils. And everybody feels that formed elements are nothing but cells. But here the very important thing is that RBC is a cell, erythrocytes, lymphocytes, neutrophils. These platelets are cell fragments. Do you remember this one? Cell fragments are formed from cell called as a megakaryocytes. Actually, in the process, these megakaryocyte is a cell which is split into cell fragments. Our cell fragments we are calling as a platelets. That is why in the formed elements, platelet at any platelets you can call, thrombocytes also you can call. ఈ platelets, thrombocytes not at all corresponding to, they are not belongs to the cell fraction. I hope you can understand. Platelets or thrombocytes are belongs to cell fragments formed from the splitting of cell called as a megakaryocyte. That is why platelets we never consider as a cell. Remaining okay. The next question, sir. The next question is find the incorrect statement. Now this is the sixth question. This is your term. Okay, your term. Just you can carefully observe. Find the incorrect statement. Almost the persons we can see. Almost you can see everything is correct only. Okay, one time you can check. Neutrophils and monocytes are phagocytic cells. Correct. Okay, that will make the explanation. Mast cells in the blood, heparin, are involved in the inflammatory reactions. Eosinophils resist the infection and also associated with allergic reactions. As the statement. A reduction in the number of platelets that leads to excess loss of the blood. These are the four options you are seeing. Among the four options, which option you feel is an incorrect statement? Incorrect statement. Actually, now I will tell you, wait for that option. Okay. So, here RBC function, for example, if I take RBC responsible for the transport of the oxygen and carbon dioxide. It's okay, right? So, RBC responsible for the transport of oxygen and carbon dioxide. If I say RBC mainly responsible for transport of, what is your option? In general, RBC responsible for transport of both oxygen and carbon dioxide, as we know. If I ask the question, RBC mainly responsible for the transport of, mainly, go for the oxygen. Because 97% of the oxygen transported by the hemoglobin, the RBC or RBC, just up to 20 to 25% only RBC, carbon dioxide transported by the RBC, because majority of the carbon dioxide transport in the form of the bicarbonate. That is why RBC mainly concerned with the transport of the oxygen. That's okay, RBC okay. Now comes to the WBC. Now you can go for the neutrophils. Neutrophils. Next one is called as a eosinophil. Just I am writing. Next one is a basophils. I wrote the what it called as a granulocytes. A granulocytes I am writing is a monocytes. Monocytes. Next one is a lymphocytes. These are the blood cells. Platelets you can put one side. Okay. What is the function of neutrophils? If you remember neutrophil, there is other famous name for the neutrophil. That is called as a PMNL, polymorphonuclear leukocyte. Do you remember that? The same thing is present in the cellular barrier of the human health and disease. So, neutrophils are also called PMNL, polymorphonuclear leukocytes. Okay, neutrophils. These function wise directly, neutrophils are the phagocytic. Here I am telling all these are related to the blood. Next one is called as a eosinophils. Directly they mention resist infection. Resist infection and also associated with the allergy. That's important. Okay, functions important. Basophils release as the blood. Okay, basophils release as the histamine and serotonin involved in the inflammatory reactions. Inflammatory reaction. Up to here, I hope everybody clear. Okay. Next comes to the monocytes. Monocytes also phagocytic. Monocytes also belongs to phagocytic, just like a neutrophils. Next one is the lymphocytes. So just here, the lymphocytes directly I can write. Lymphocytes are play important role in the immune system. That's very good. Just I'm asking. So those who are seeing, how many types of lymphocytes are present? Total lymphocytes. Anybody? How many types of lymphocytes are? How many types of lymphocytes are present in our body? Types of lymphocyte. Generally, student will tell the B lymphocyte and the T lymphocyte. Correct na? B lymphocyte and T lymphocyte. Anybody, can you mention in the what it called as a comment? Can you mention any other type of the lymphocyte? Any other type of lymphocyte? Can you fastly type the answer? Lymphocytes play important role in the immune system. Okay. What are the types of lymphocytes? B lymphocyte, T lymphocyte. One more important lymphocyte is a natural killer cells. Natural killers. So why we are forgetting, B and T lymphocyte, they are part of the specific immune system. Natural killer cells are part of the non-specific defensive system. That is the innate immune system. They are present in the cellular barriers. That is why usually natural killer cells immediately you will forget the type of lymphocyte given in the NCERT. Okay. As we know, B lymphocytes mediating the HMI. What is HMI? Humoral mediated immunity. T lymphocytes mediating the CMI, cell mediated immunity. Natural killer cell, already I said, they are part of the cellular defense mechanism. Cellular defense mechanism. This is a overall. One time you can check. Nice one. Here it is a thing is a RBC function. Among these one, overall information is very important. Okay, na? When go to the exam hall, the confidence that leads. Okay, neutrophils phagocytic, eosinophils resist the infection and associated the allergy. Basophils secretes the histamine and serotonin inflamed in the allergic reactions. Monocytes are phagocytic, lymphocytes immune system. That's okay, very good. So percentages you came to know, followed by the functions. Okay. Now in this one, what is the wrong means? If you observe carefully here, the thing is that second option one time you can check. Here I mentioned the mast cells in the blood. Practically here basophils, I hope you can. Basophils is equals to mast cells. You remember this one? Basophil is equal to mast cells. What is the difference between these? Basophils present in the blood. Function wise, mast cells, basophils both same to same. The difference is mast cells present in the tissues. You remember? You got it. So, for example, if the question is a framing like this, which cells produces the histamine serotonin in blood involved in the inflammatory reaction, going for the basophils. Which cells produce the histamine and serotonin in the tissues, produces the inflammation reaction is a mast cells. So, functionally, mast cells, basophils, both are same to same. Only difference is the location. Small point. It gives us so much complex flexibility in the concept. Same point I am going to tell. Next one is a monocyte. Is a phagocytic cell, already we know. That is equals to macrophage. Only difference is monocyte is a phagocytic cells present in the blood. Macrophage is a phagocytic cell present in the tissues. Same difference between the, what is the difference between the basophils and mast cells? Same difference you can observe between the monocytes and macrophages. That's good. It's not only Kamali Palani. It's not only in the areola tissue, madam. That's good. Mast cells in the areola tissue is good. But thing is that these mast cells not only present in the areola tissue. It's present in the every tissue present. Because wherever in our body other than the blood, wherever inflammation other than the blood in the body, wherever the inflammation reaction occurring, Wherever histamine and serotonin is releasing in the tissue in our body, that is produced by the mast cells. That is, I hope you can understand. That's very good. Okay, na? Anyhow you said. So, mast cell is equal to basophil. Only difference is the blood and tissues. Next one, monocyte is equal to macrophages. Both are functionally phagocytic cells. These monocytes present in the blood, macrophages present in the tissues. I hope that composition of the blood, you are okay. Now I am going to the next question. Okay. The next one. This is a very important concept in the body fluids and circulation. Body fluids, and one is erythroblastosis fetalis, very highly conceptual, followed by the cardiac cycle. So this is a directly I frame the statement. Erythroblastosis fetalis is due to, anybody can you comment the answer? Erythroblastosis fetalis is due to which incompatibility? That is a RH incompatibility. Direct answer, very important, highly conceptual. So here directly I am telling only one statement. Here we cannot discuss the total process. RH erythroblastosis fetalis is due to RH incompatibility. Everybody, can you somebody comment? In erythroblastosis fetalis, mother, RH positive or RH negative? So very good. Can you make it? Mother is RH positive or RH negative? Here in case of if erythroblastosis fetalis to induce, mother is a RH negative. RH negative. I hope you can understand. And the fetus is a RH positive. RH positive. This mismatch, very important concept, okay, no doubt about it. This mismatch you can call it as incompatibility. That is why here I am telling this one, erythroblastosis fetal, RH negative, good. This one is Aishwarya, Maheshwari, that's very good, okay, na? Like that. So, not only answer, when you are telling that question, bring the concept around it. Okay, automatically when you attempt the question, automatically you will come to know that one. Okay, so I am concluding 7th question, erythroblastosis fetalis is due to RH incompatibility or RH mismatching. 1. In case of erythroblastosis fetalis, mother is RH negative, so fetus is RH positive. Okay, the next question I am going to give. 8th question for you. Can you just look and tell? Good, Balu. So, Balu Sri. Okay, good. So, can you check the 8th question? Erythroblastosis fetalis can be avoided by administrating. Options you can see and give the comment. DRH, there is a deep level. I said erythroblastosis fetalis is highly conceptual. Okay, so further levels you can read in the graduation. Okay. So, can anybody, question number 8, by administering the anti, option 1, 2, 3 and 4. A small difference is only there. Can you check and tell me? Can you check and tell me? This is which question number also you need to mention, please, okay na? Okay. So, yes, good. Maheshwari, Balu Sri, okay. And Kamali Palani, you mentioned 3, one time you can check, don't make it any thing. Erythroblastosis fetalis is avoided by giving the anti-RH antibodies, not to the fetus, no need to give the fetus, okay na? First option, third option I am deleting. Giving the anti-RH antibodies to the mother immediately after the delivery of first child, or can mother 48 hours after the delivery of the first child? Definitely option 2. If you are telling this option 2 means you are conceptually good. So, I am telling erythroblastosis fetalis can be avoided. That's good. Make the question number also, then I will get it. 8th question I am telling. So, erythroblastosis fetalis can be avoided by administering the anti-RH antibodies giving to the mother immediately. That's very nicely concept, you know, immediately after the delivery of the first child. Because in the erythroblastosis fetalis affecting person is the second child. That is here, because of my limitations of the questions, erythroblastosis fetalis, I covered only 2 questions. One is erythroblastosis fetalis is due to the RH incompatibility. That means mother is RH negative, fetus is RH positive. Second point is erythroblastosis fetalis can be avoided by administrating, can be prevented by giving anti-RH antibodies to the mother immediately after the first delivery. Okay, going for the next one. Okay, what is this anti-RH antibodies, that we can study. Okay, next one, I am going to the next question. This is also important question, exam point of view. This is important question given in NEET exam lo 1 year given question, directly we can make. So ninth question, okay, which enzyme can convert fibrinogen into fibrin? Here 4 options are there: thrombokinase, thrombin, hexokinase, succinate dehydrogenase. Ninth question. Ninth question. Can anyone, what it called, Balusri? Next one is Aishwarya. Next one is a Maheshwari. Can you give the ninth and tell me? That's important question. Okay, our blood clotting paragraph is a very small paragraph. It's very important, conceptual important, exam also important. Okay, I hope I gave the 2. Okay, that's one. Good. So that is a ninth question too. That's now I am going to give this one. Good. Kamali, that's good. 9-1 is a 2. Important question itself. First, directly we can go. So here I am telling, when the tissue damage happened, or you can call it as a blood damage happened, it initiate the cascade reactions. As a result of cascade reaction, there is activation of an enzyme is called as a thrombokinase. First I will cover the question, then I will go for the options. Okay. So tissue damage or blood damage happened, that is to activate the cascade reactions, extraordinary word, cascade reaction. As a result of cascade reaction, there is activation of an enzyme complex is called as a thrombokinase. This thrombokinase is a kinase you can call it as a prothrombin activator also. So you can call it as a prothrombin into thrombin. This thrombin is a converting the inactive, that is a fibrinogen to fibrin. I hope what is there on the screen, what is there on the slide, you can see. Okay, na? So tissue damage or blood damage happened, it activates the cascade mechanism. That cascade mechanism activates the enzyme complex is called as a thrombokinase. Thrombokinase converting the prothrombin into thrombin. This rate determining step. Rate determining step of the blood coagulation is this conversion, prothrombin into thrombin only. That thrombin is converting the prothrombin into fibrinogen into fibrin. Simple, 2 reactions. So I am going for option 1 I covered, option 2 I covered. Now I am going for the hexokinase. Anybody, can you comment? What is the reaction mediated by the hexokinase? Anyone? Can you comment hexokinase reaction? Hexokinase reaction. First enzyme in the glycolysis, hexokinase. Anybody, can you comment? Hexokinase reaction. Come on. Hexokinase. I hope I didn't remember exactly how many kinases. First here I am calling as, first I am going to clear the thrombokinase. Thrombokinase converting the prothrombin into thrombin. Second one. Hexokinase is converting the glucose. Remember the first reaction. That's good, Aishwarya. Glucose convert into glucose 6-phosphate. First reaction. Extraordinary step, you know, when you get into the graduation, first year biochemistry. So this reaction you can study like anything. So second, first one thrombokinase, I said hexokinase, first enzyme of the glycolysis. That is a glucose convert into glucose 6-phosphate. The next one more kinase is there, if you remember, streptokinase. Streptokinase. Can somebody type and tell what is that's streptokinase? Tell something. So 3 kinases. thrombokinase, hexokinase, streptokinase. Anyone can you type streptokinase? If you remember, the chapter is the microbes. In that chapter, you will get the streptokinase. Extracting from bacteria is called as the streptococcus. Anybody? Yes, yes, very good. Aishwarya is good. So this is a clot buster. Clot buster. Class buster. So that class buster used in the treatment of the myocardial infraction that leads to heart attack. Extract three kinases. That's good. Three kinases we are telling. One kinase is a hexokinase, glucose to glucose 6-phosphate. Thrombokinase, which converting the prothrombin into thrombin. Streptokinase, extracting from the bacteria streptococcus. That is a recombinantly produced streptokinase used as a clot buster, used in the treatment of a myocardial infraction that leads to heart attack. Next one, anybody can you comment the succinate dehydrogenase? Streptokinase true to so yes, myocardial infraction. Good. Aishwarya is a good. Next one is can you go for succinate dehydrogenase? Absolutely, this enzyme in the NEET exam 3 times. What I remembered only 3 times given question on the succinate dehydrogenase. Anyone? Can you comment what it called as here? Along with the question. Question already I said absolutely important. Okay. Options. Succinate dehydrogenase. Can anyone succinate dehydrogenase? You can see in the what it called as succinate dehydrogenase. 2 chapters you can study succinate dehydrogenase. Good ma. That's chapter name and tell succinate. What is the reaction carried by the succinate? It is a part of Krebs cycle. First time telling is a part of Krebs cycle. Good. No. Aishwarya, this you made another way, okay? Is a substrate level phosphorylation. No, that's not the correct. Succinate. This one is you made succinate dehydrogenase. This is involved in the what it called as a Krebs cycle that converting the succinate to fumarate. Succinate to fumarate. Sorry. Succinate to fumarate. Here the coenzyme is FAD. FAD converted into FADH2. This is not SLP, madam. Okay, na? It's not SLP. Succinate dehydrogenase, that is a part of the Krebs cycle. Very important, you know, this enzyme. Very important. So, how much important? Just give me. First time telling, succinate dehydrogenase enzyme is a part of the Krebs cycle which is occurring in the mitochondrial matrix. Are you clear? That converting the succinate to the fumarate, succinic acid to the fumaric acid, that producing the FAD into FADH2. 1 point. Second important point is that succinate dehydrogenase. What is the competitive inhibitor of succinate dehydrogenase? Competitive inhibitor of succinate dehydrogenase. This you can mention. In our whole syllabus, only one competitive inhibitor is only present. What is the competitive inhibitor of succinate dehydrogenase? Anyone? Give the first comment on it. I will read the name. First comment. What is the competitive inhibitor of succinate dehydrogenase? Good. Aishwarya. So, that one is a malonate. Good. Malonate. Malonate is very important, you know. So, malonate or malonic acid, you can call. Malonate is the competitive inhibitor of enzyme succinate dehydrogenase. Second point. Third point, if you remember the electron transport system containing complex 1, complex 2, complex 3, complex 4 and complex 5. Among these 4 complexes, you want secret, complex 2 is nothing but succinate dehydrogenase. What a greatness of the evolution, you know. Succinate dehydrogenase is a one enzyme which is a common, same enzyme which is a common in both Krebs cycle and also electron transport system. So that is why, it having its own integral value. That is why 3 points I covered. Okay, that's good. Complex 2, getting on. Pranav brother, you are telling getting only 300 in bio. we need to increase. Okay. So we need to increase. 300 is not enough in the bio. Okay. Maximum probability. So I am telling those who are Pranav, maximum probability in the biology, you need to get wrong answer probabilities, either 1 or 2%. 1 or 2%. That's only possibility. Because we did work from June to December, almost January first week we completed the syllabus. Okay. So that is why 1 or 2%. Okay. That's all only. Okay. If the biology law, the probability, if you are making 300, 290, 310, it's not enough. That's why I'm telling, I'm going forward. 9th question, option 2 is a thrombin, thrombokinase I said, hexokinase I said, very important succinate dehydrogenase, 3 point I said. Okay. Going for the next, very important sir, succinate dehydrogenase. Okay. Next one, 10th question. Dash ion play important role in the clotting. Direct question. This is a very important. Okay. Na? Blood clotting already we said. So this is a why question is important. I will tell you. Okay. First, can you comment the answer? Which of the following ion play important role in the blood clotting? Blood clotting. Option 1, if you can't see properly, magnesium. Second one is a calcium. Next one is a copper ions. The next one is a magnesium ions. Anyone? So very good. Pranav, that's good ma. Calcium. So calcium is a essential, is a very important for the blood clotting. Can you? That's very good. That's very good. Here, what is a repeater sense? I am going to tell. Calcium I am going to take. Remaining options I will take soon. Calcium I am taking. I will tell you one by one. In what are the chapters calcium is there? First I am telling calcium is absolutely required for the blood coagulation. Blood coagulation you can call, or you can call it as a blood clotting also you can call. Blood coagulation or blood clotting. Okay, 1 point. So calcium, here I am writing calcium. Calcium is required for the, absolutely required for the blood coagulation. Now I am telling, calcium is required for the muscle contraction. Calcium is required for the muscle contraction. Anybody, can you tell? Yes, wow, very good, very good. Aishwarya is very good. So it's a part of this. Thank you very much, Aishwarya. So third point is not my point. This is said by the Aishwarya NR. Calcium is a part of the middle lamella. That's very, thank you very much. Is a part of middle lamella. Middle lamella. This middle lamella main component is a calcium pectate. This I know, but I forget practically. So third one, thank you. The next one is any other. Blood coagulation, muscle contraction, no. I need to explain the options. I need to explain the options. Okay, wait. Don't go to the next question. Okay, that's a time schedule. So time will be there in the final exam. Okay. So calcium is required for the blood coagulation, muscle contraction, middle lamellae, calcium pectate, any other function. Now I am telling, calcium is requires to induce competency. Induce competency. Do you remember the induce the competency? Anybody remember this one? So induce the competency in biotechnology. Biotechnology 1. Before a host cell is a subject, before a host cell is subjected to the transformation, the host cell must induce the competency. Competency is induced by treatment with a specific concentration of calcium. Specific concentration of the calcium. That's one. Any other function of the calcium, do you remember? Calcium. The calcium along with the IP3, inositol triphosphate, acts as a secondary chemical messenger. You remember this one? Cyclic AMP is a one secondary chemical messenger along with the calcium and IP3. So see, this is called repeaters. This is called repeaters. Pranav sir, this is called repeaters. How to study? Calcium, when we are telling the calcium involved in the blood coagulation, required for the muscle contraction. Thank you, madam. This is a middle lamella calcium pectate. Calcium is required to induce the competency. Calcium along with the inositol triphosphate acts as a intracellular secondary chemical messengers. Along with this one more. Calcium present in the seminal plasma. Calcium present in the seminal plasma supports the sperm motility. This is a way of reading. This is a way of reading. Calcium. This is whoever read like this, the person is a repeater. So this is, I said, comes to the magnesium. Any enzyme in the glycolysis, Krebs cycle, any enzyme, magnesium is a metal ion. That is a pyruvate dehydrogenase. If you remember, pyruvate dehydrogenase require metal ion is a magnesium. Okay. So magnesium is a metal ion for the so many enzymes. Here I am telling in our syllabus pyruvate dehydrogenase. Next, anybody tell the copper ions? So these copper ions electron transport, which complex? I am arising the question. Which complex? Among complex 1, complex 2, complex 3, complex 4, among 4 complexes, which complex in the electron transport system containing a copper centers? Anybody, can you comment? Among the 4 complexes, which complex containing a copper centers? Cu, copper centers. Anyone? Can you please comment? Fast. Good. Complex 4. Copper ions. That's good. I asked a question. Okay, na? Complex 4. Complex 4. 2 copper centers are present. CuA, CuB. So that is a 1 point. It's a complex 4. Good. Complex 4. Second very important thing is that this Pranav mentioned. This one is copper ions. In case of the copper T's, copper ions, calcium is required for the sperm motility. This is copper ions suppress sperm motility. Suppress sperm motility. These two are regarding the copper ions. Good. Next one is iron. Iron is a very important. It is beyond my explanation. Iron is very important. Is a part of cytochromes. It's a part of cytochromes. Do you remember the word is a cytochromes? Cytochromes are, they're extraordinary products. So they are involved in the electron transport system. So you can observe dash along with the question. What are the remaining things you can observe? Okay. Now I am going to the next question. Okay. Next one is, among the following statement about human heart, find the incorrect one. Direct question only. Important question. Important question. Match the following. Look, given these options given as match the following in the 1-year NEET exam. The right atrium and the right ventricle is guarded by the tricuspid wall. Okay, right? Left and what it called as a left atrium and left ventricle is guarded by the bicuspid or a mitral wall. The opening of the right and left ventricle into the pulmonary artery. Into the pulmonary artery. One is 11, one is 2. One more time, can you check Balu? Ball is 3. One more time, can you check? The first and its 2 options are very important. Okay, na? The opening of right and left ventricle into the pulmonary artery and aorta provided by the SLV, semilunar valves. Fourth option, the valves in the heart allow the blood in the both direction. It is wrong. That is wrong. It allows the blood, these valves, if you make just a hypothetical diagram, this is a right atrium, this is a left atrium, this is a right ventricle, this is a left ventricle, okay, with corresponding artery. This is a pulmonary artery, this one is a aorta, this is called as a tricuspid valve, this is a bicuspid valve. Here you can find the semilunar valves. So, without prolonging, I am telling, the two statements are very important. Always there is a confusion. Tricuspid valve, bicuspid valve, always confusion. Please, one time you can check. Okay. These valves in the heart allow the blood in the unidirectional flow. Blood always flow only from the atria to ventricles. From ventricles to the next corresponding arteries. Only unidirectional. It never be from arteries to the ventricle, ventricles to the artery. Blood cannot move. That is why unidirectional flow only. arteries to the ventricles, ventricles to the corresponding arteries. That is why I am telling 11th question option 4. Okay. Next one is this one is a 12th question. Can you check and tell me? This is a somehow I thought of framing. So, sinoatrial node. Where is the sinoatrial node is locating? Upper right corner. If you take a rough structure like this, if you take the right structure, this is a right atrium. Upper right corner of the right atrium you can see. That you can see the sino. Why it is called as a pacemaker? Why it is called a good Pranav? Why it is called? This 12th one is a 3. Bolus 3. Okay, Pranav. One time can you check ma? San and the people will get confused. Here that overall statement is give one fraction I will complete. Here in the heart nodal tissue we say one is called as a SAN. Next one is called as a AVN. Next one is called as a ventricular, that is right and left muscle bundles along with the Purkinje fibers. This is called as a ventricular myocardium. I hope you can make it. One is a 3 nodal tissues of the heart divided into like understand into 3. One is the SAN, next one is AVN, next one ventricular myocardium. SAN produces the action potential 70 to 75 times. AVN produces approximately 50 times. This is the produces approximately 20 times action potentials. That is why the NCERT law there is a statement. Number of action potential generate varies from the one part to another part. But why it is called as a SAN is called as a pacemaker? Because the SAN among the SAN, AVN, ventricular myocardium, because SAN produces the maximum number, it only produces wrong. All will produce as the action potential because SAN produces the maximum number of action potential, 70 to 70. That is the reason why SAN is a pacemaker. SAN is a pacemaker because not only it produces, no, it produces maximum number of action potentials. So, that is I am going for option 2. Please make it to small confusion. Only produces, no. It produces the maximum number. Okay, going forward. Next question. There is no much explanation. The duration, because it's highly conceptual. How much I say also something will be remain. Duration of the cardiac cycle 0.8 second. Option 2 I am going 0.8 second. Out of this one, can you comment somebody? Atrial systole, what is the time duration? Ventricular systole, how much time duration? Joint diastole, how much time duration? Can you check? Good, option 2, that's okay. Direct answer. Can you check? I am asking, what is the time duration for the atrial systole? What is the time duration for ventricular systole? What is the time for joint diastole? Joint diastole. Can anyone, can you please make it? Time duration for atrial systole. AS you can call while typing. VS you can call. JS you can go. Joint diastole. JD you can call. Anyone? Fast. Pranav. Safana. That is Dr. Safana. Okay. The next one. Joint diastole. Pranav, can you check ma one time? Can you check one time? Anyone? Anyone? Aishwarya? Can you check? What is the time duration for atrial systole, ventricular systole, joint diastole? Who won? Atrial systole. Can I go for the answer? Atrial systole time duration is a 0.1 second. Atria is a thin. Ventricular systole it will take the 0.3 second. Joint diastole 0.4 seconds. 0.4 seconds. Okay. Atrial systole, this is a together 0.1, 0.3, 0.4 together is a 0.8 second, 0.8 second. Okay. So 13th question directly I am making, okay, going forward. In ECG, this question is also, this question, it's not about the question, it's about the concept. ECG is, that area is important, one question possibility, okay. So in ECG, as we know, there are like P wave, next one is a QRS wave, next one is this is a P wave, QRS wave, next one is a T wave. P wave represents the atrial depolarization. Good. Atrial depolarization. That's important. Three important. Okay, na? Atrial. QRS wave indicates a ventricular depolarization. Ventricular depolarization. T wave represents the ventricular. Sorry, then is this one is, it represents the ventricular repolarization. These 3 points important, okay na? Good. So this one is a P wave represent the atrial depolarization, QRS wave represent the ventricular depolarization, T wave represent the ventricular repolarization. Okay na? This question is a particular small area. So that is important. Okay na? Now next time going. Which of the following change? This is a for you. So you can make it. 15th question is for you. The following change is not represented in the ECG. Anybody, can you represent? Good. Balu. 15th question. The following change is not represented, is not expressed or not represented in the ECG. That one. Anyone? Good. Prem. That's good. Aishwarya, good. So actually, P wave, just now I said this very nice thing, you know. P wave represents the atrial depolarization, QRS wave represents the ventricular depolarization, T wave represents the ventricular repolarization. But where is atrial depolarization is for ventricles, ventricle repolarization is there, ventricular repolarization is there. But for atria, only atrial depolarization is there. There is no atrial repolarization in the ECG. Okay, that is I am going for in the ECG. Nice concept. Option 2 I am going forward. In ECG, there is no representation, there is no indication of the atrial repolarization. Because when the atria is undergoing repolarization, atrial repolarization is a coincidence with ventricular systole. You understand now? When the atria is undergoing repolarization, ventricle is undergoing the depolarization. That is the reason why there is no representation regarding the atrial repolarization. So, good, Radha. That's good. Second one. The next one is, this is important only. Okay, we'll make it. So, is a conceptually important. Okay. A unique vascular connection exists between the digestive tract and liver. Direct answer. This is between this one, option 3 I am going. Okay, option 3, this I am going is a hepatic portal circulatory system. You remember these three? Renal portal system, hypophyseal portal and hepatic portal system. First we will speak about the hepatic. Hepatic portal circulatory system is a connection, blood connection between the intestine. Between the intestine, that's good answer. Remaining option one time you can check. Remaining options one time you can check. Okay. Hepatic portal circulatory system is a special vascular connection, blood connection between the intestine with the liver. So here what happened, directly function I am telling. If you remember, once the digestion is over, the blood is moving from the intestine to the liver. Nice statement, you know. Whether you understand or not, I am not very sure, but very nice statement. Once the digestion is over, the blood is moving from the intestine to the liver before enter into before enter into systemic circulation. How many members understand this statement? I don't know. Systemic circulation. So, hepatic, so 2 points I am going to speak. Okay, na? So, hepatic portal circulatory system is a unique, so what it called as a blood connection system, which connecting the intestine with the liver, is responsible for the transport of the blood from intestine to the liver before entering into the systemic circulation. Now, somebody tell me, somebody type, hypophyseal portal circulatory system is connecting which two parts? Your turn. Safana, Balu, Aishwarya, somebody is missing, okay. So, can you come and tell me what are the two parts connecting the hypophyseal portal circulatory system? Very important. Conceptually, chemical coordination is very important. That is why he gave it. Anyone? This one is a Balusri. You were speaking about the hypophyseal portal circulatory system, na Balu? Okay, Balusri. No. Liver and no. Wrong. Hypophyseal portal circulatory. Nobody is a typing. Hypophyseal portal circulatory system is connecting. Good. Aishwarya is good. So anterior connecting from good. One is the anterior pituitary gland from good. Frame is good. Hypothalamus. There that is very important, you know, that's a chemical coordination. Now I am telling this is a hypophyseal portal circulatory system. So I will tell you the statement, understand. Hypophyseal portal circulatory system which is connecting the hypothalamus with the pituitary gland you can call, but the mostly somebody type that is a Aishwarya. That is a connecting the hypothalamus with the anterior pituitary gland. Why I said this option, you know, once the hypothalamus produces the two types of hormone, releasing hormones and inhibitory hormones. Do you remember? Releasing and inhibitory hormones produced by the hypothalamic neurons reaches to the APG, anterior pituitary gland, through hypophyseal portal vein. Do you remember? Do you remember? Why I mentioned? Last time I am telling, releasing hormones and inhibitory hormones produced by hypothalamus reaches to the anterior pituitary gland through hypophyseal portal vein or hypophyseal portal circulatory system. Renal portal circulatory system, okay, that is there in the frog. One time you can check. Okay, now I am going. That's good. Next question, wait. This is okay, generalized. Respiratory rhythm center present in the medulla. Directly I am telling a medulla. The respiratory rhythm center is mainly concerned, primarily concerned with the regulation of the respiration. Primarily concerned with the regulation of the respiration is a respiratory rhythm center present in the medulla. Okay, one. Next one, which is center present in the pons? Your turn. Which is center present in the pons? Pons have so many functions, no doubt about it. With regarding the breathing, regulation of this, which is center? Your turn. 17th one, that's good. Answer already I said, okay. There is a small time gap between me and that telecasting, that one. One time can you check? Pons region. Pons region. Good. There's a new person added, Anbalgam. Anbalgam. So that's very good. That is a pneumotaxic center. Good. Aishwarya is good. Pneumotaxic center. Somebody deleted that one. No problem. Even if you say wrong also, there is no issue. So pneumotaxic center which is present in the pons. This is what it called as this pneumotaxic center reduce duration of inspiration. Do you remember? Inspiration or inhalation reduces the duration of inspiration or reduces the duration of the inhalation and alter the breath rate. That is the one. Anybody tell? That's very good. Cerebrum, okay. All function. That is doing by the cerebrum only. No need. Seat of intelligence. That's nothing but cerebrum. The exam point hypothalamus. There are, you tell, at least 3 functions of hypothalamus. 3 functions of hypothalamus. Tell, I will announce the name. Three functions of hypothalamus. Anyone? Aishwarya, Kamali, Palani, Prem, Dr. Safana, Balu, Radha. Yes. Just to tell the three functions of hypothalamus. Fastly type. Definitely, always there is a constant question in the hypothalamus in the NEET exam. Always. Always hypothalamus lo that question constant. Tell. Good. Venusri, that's good. That's good. Maheshwari, good. Aishwarya, good. You said the hypothalamus control. Yes. This absolute. Control the body temperature. I said three functions. Body temperature. That's good. Venusri. That controls the hunger and thrust. Hunger and thrust. Any other? You forget about one very so that's good. Aishwarya, Maheshwari is good. Control the body. This is a damn sure question in the NEET exam. Damn sure. Any other? One is hypothalamus is a body temperature, control the hunger and thirst. Any other? It is osmostat. Osmostat. Go and check the regulation of the excretion. Bah, extraordinary. Osmoreceptor switch on, osmoreceptor switch off, release of the ADH, if you remember. Okay? So, in that context of osmostat, any other function? Good, Kamalni. Hunger, thirst, thermoregulation. One more very important. Along with the limbic system, hypothalamus controls the sexual behavior. Sexual behavior. If you remember, 4 points. Once you take the question paper, among the hypothalamus, one question. So, hypothalamus is controlling the body temperature, controlling the hunger and thirst, controlling the water volume, that is called as a osmostat. It along with the limbic system, the hypothalamus involved in the along with the hypothalamus. Sorry, that statement is it went wrong. Along with the hypothalamus, limbic system involved in the regulation of the sexual behavior. Okay, good. Thermoregulation. Thermoregulation is one of the function. Going forward, okay. Respiratory, I am concluding the question. 17th question I am going to conclude. Respiratory rhythm center is a primary concern with the regulation of the respiration. Present in the medulla oblongata. Pneumotaxic center, which is a switch off point, reduces the duration of inspiration, present in the pneumotaxic center. Chemosensitive areas also present in the medulla. 3 centers. I am telling 3 centers. One is a respiratory rhythm center present in the medulla. Chemosensitive areas present in the medulla. Pneumotaxics present in the pons. Okay. Now I am going to the next question. Hypothalamus question will come. Next one. This is the question. If anybody tell this question, I am giving 18th question. I am telling to everyone who is there on online. You can see also. Once I said the answer, no X. So, before going to tell only, I am telling the answer. Whoever is there in the line now, Maheshwari, Aishwarya, Venusri, Kamalni, Balu, Dr. Safana. Safana is missing now. I think she went to take the coffee. Can you wait for me? What did it called as 10 minutes? Okay. Can you come on to the online? This one. Anyone? Good, Aishwarya? Move on. Anbalgam. 1. Dr. D.K. Tamil. Balusri. Tell, answer you tell. Aishwarya is good. Aishwarya is good. Aishwarya, I think. Oh, so many Aishwarya. Okay, Aishwarya changed the option from 4 to 1. Option 1. One time you can change. This is I intentionally gave. Radha, yes. I can understand. So, no one. Other than no one. Maheshwari, there is no answer from you. Can you check? This is why this question I gave you, no? Just you can observe. Okay, no? Once explanation, rather. 1, no. That is I said, everybody that one remembered, there is a note, sir. That one is a clear statement, sir. 1 here, the question is 1 RBC or 1 hemoglobin, sir. Just one question, sir. 1 hemoglobin carry. So how much what it called as a gap is present, you can observe. Just let me give time. One hemoglobin molecule carry maximum of 4 molecules of oxygen. Correct na? 4 molecules of oxygen. One hemoglobin molecule, sir. One hemoglobin, no, wrong. No, it's not the first option. You can check the question properly. It's not about the one hemoglobin, it's one RBC. Intentionally I gave. Okay. One hemoglobin molecule carries a maximum 4 molecules of oxygen. Yes or no? But now you can take one hemoglobin molecule means, are you sure? Alpha 2, beta 2. First let me, okay, na? I will clarify, then I will go for the hemoglobin. Okay. Sir, if you take one RBC, only one RBC you take, one cell you take. In this RBC, there is no nucleus. You know, right? Correct, na? There is no endoplasmic reticulum. There is no Golgi complex. There is no mitochondria inside, cytoplasm I am speaking. There are no vacuoles. Nothing inside, sir. Cytoplasm of the RBC just empty. There is no nucleus, there is no ER, there is no Golgi complex, there is no lysosome, there is no mitochondria, nothing. Then what is there? Cytoplasm of the RBC consists of maximum number of hemoglobin. I am telling difference you can understand. One RBC cytoplasm containing maximum of 200 million hemoglobin molecules. One RBC consists of approximately 200 million hemoglobin molecules. Now you can differentiate. One RBC carries how much difference, how much you made wrong, you can check. How much this one? One RBC containing 200 million molecules, 200 million molecules of hemoglobin. That means one RBC carries how many? 200 million into 4 oxygen molecules. That is why, sir, there is a small difference is there between the reading and understanding. Please understand. Okay. That is why I am telling here I am going for option 4. So how many members to accommodate? Yes, Venusri. How many members understand this explanation? One hemoglobin molecule carries the maximum of 4 molecule of oxygen, not one RBC. How many members understand? Can you give the thumb on the comment box? One time you can make it. One, can you give one thumb on the this one? Aishwarya, thank you. Anyone? Relaxing video, the nice title. Yes, Radha, thank you. Any other? Can you give fast? Thank you. And there is a difference. Okay, here a small discussion before going to tell this one. Before 90th question is a very good question. Exam point of view. Hemoglobin, that's HB, hemoglobin. Thank you to everyone. So hemoglobin, a few points I will tell you, then we get inside. Okay, hemoglobin is represented by alpha 2, beta 2. Everybody know, because that's, we are damn sure alpha 2, beta 2 means there is 4 polypeptide chains are present, 2 alpha polypeptide chain, 2 alpha subunits, 2 beta subunits. Okay, each subunit carries the 1 molecule of oxygen, so it carries the maximum 4 molecules of oxygen. Okay, hemoglobin, 1 point. Okay, hemoglobin is represented by alpha 2. Hemoglobin molecule is the only example in our syllabus exhibiting the quaternary structure. Remember this point? Hemoglobin is the only example present in our syllabus which exhibits the quaternary structure. Hemoglobin. This one is, can you correlate with the sickle cell anemia and tell me what is the statement? This sickle cell anemia is due to point mutations. Yes or no? Point mutation. If you remember sickle cell anemia, glutamic acid is replaced by the valine at the 6th position of beta polypeptide chain. Do you remember this word? Hemoglobin related. Hemoglobin related. Okay. In sickle cell anemia is due to point mutation. In sickle cell anemia, one codon will be changed. That results glutamic acid is replaced by the valine at the 6th position of beta polypeptide chain. Next one, thalassemia. Thalassemia. If you remember, thalassemia is due to the deletion mutation. The pen is getting slow. Okay, sorry. Yeah. It is due to this one. So, fetal RBC, how many alpha? Brother, this pen is not working. Brother, what do you call? Anna Balan. Sir, I will tell you, fetal RBC, sir, hemoglobin alpha to beta, it is related. In the fetal vanda, just I am giving the clarity to the person. Fetal, good. Venusri, that's good, ma. The alpha to beta 2 is related to the adult. In case of hemoglobin, it is called as HbF. HbF is represented by, pennies are slow, getting slow, alpha to gamma 2. Alpha to gamma 2. Anyhow, in this question, thank you, Anbalam. Anyhow, you gave the good concept. Just I'm asking, genetic disorder. Just get ready to comment. Make it a comment. Sickle cell anemia is a genetic disorder. Okay. So, transmitting from the parents to the offspring. Fetus, does the fetus suffer with the sickle cell anemia or not? Say yes or no in the comment. Does fetus suffer with sickle cell anemia or not? Yes or no? Your turn. Fastly tell. This point just I will correlate and I will go normally. Good. Venusri, move on. Yes or no? That's enough. No, that's good. Aisha, nice. No. Kirtana, wow. New member. Next one. Hi Aisha, hi Kirtana. Kirtana said yes. Aishwarya said no. Harshini said no. Any other? Fast. Sir, very small difference. Sickle cell anemia, just I said, sir. What did it called as unbalance? You didn't comment, brother? Just one time you can check. Sickle cell anemia, what happened? Glutamic acid is replaced by valine at the 6th position of beta polypeptide chain. Beta polypeptide chain. Practically, there is no beta polypeptide chain in the fetus. Fetus low, alpha to beta 2 no. Alpha to gamma 2. Alpha to gamma 2. You understand? Even though sickle cell anemia is a genetic disorder, fetus will not suffer with the sickle cell anemia because in the fetus hemoglobin there is no beta polypeptide chain. That's good. Thank you. The next one is a thalassemia. Thalassemia is due to the deletion mutation. There is a thing. Here in thalassemia, alpha thalassemia, beta, there is a reduced synthesis of either alpha polypeptide chain or beta polypeptide chain. So, all four concepts are related to the hemoglobin only, be confident. Hemoglobin is represented by alpha to beta 2. One hemoglobin molecule maximum carries the 4 molecules of oxygen. Hemoglobin is the only molecule in our syllabus exhibiting the quaternary structure. Next one. In the sickle cell anemia, glutamic acid is replaced by the valine at 6th position of beta polypeptide chain. It is due to the point mutation. Thalassemia is due to the, what it called is a deletion mutation, involves a reduced synthesis of either alpha chain or beta chain. The last statement is a very nice point. Sickle cell anemia is a qualitative anemia. Thalassemia is a quantitative anemia. Sickle cell anemia is a qualitative anemia. Thalassemia is a quantitative anemia. Now I am going forward. Okay. This one is a. This one we can go forward. Okay. That is we are going to test chromosomal analysis. In amniotic fluid, if you observe, we are not going to test the hemoglobin molecule. We are going to speak about the gene. Whether the gene is we will compare with I am speaking to How is that possible? Kirtana I am speaking. Kirtana madam, in amniocentesis, we are going to see the gene of that hemoglobin molecule, whether it is a functional or not. Here we are not going to speak about, here we are not. This is a hemoglobin molecule, it's a protein molecules present within the RBC. In amniocentesis, we are going to check whether the hemoglobin gene, whether it is a normal gene or is it under abnormal or mutated gene. That is why. Okay, we are. So, we can make. Next question I am making. Binding, this is exam point of important. This is directly I will tell you, by exam important. Binding of oxygen with, this is a your turn, this I forget, okay. So, anyone, binding of oxygen with hemoglobin is a primarily related to, 19th question everybody. This is I made for you only, one time. Anyone, Balu. Fourth option, very good, move on. Question one time can you check? Question one time can you check? Binding of oxygen with hemoglobin is a primary related. Two questions I give like this. One time you can check. What it called as a Balu, fourth option. Aishwarya, fourth option. Kirtana, fourth option. Anbalgam, fourth option. Venusri, fourth option. Somebody missed. Radha is a missed. Anyone? Who is this? Aishwarya. Sir, here, there is a, see, these almost Aisha, first option. That's good. Ma, here, you can one time you check the NCERT. Well, I will clarify, just one time you can check. Binding of hemoglobin here primary related to, only one option is a partial pressure of oxygen. Statement I am telling you can check. Binding of oxygen with a hemoglobin is a primary related with a partial pressure of oxygen, full stop. P, partial pressure of carbon dioxide, H plus ion concentration, body temperature are other factors are also affecting the binding of oxygen with hemoglobin. One time you can check. Here this one is a primary related, binding of oxygen with hemoglobin is a primary related to partial pressure of oxygen. That is why intentionally gave the all of the above option. You understand? That's why. Okay, going. Next question important exam ki. Directly I am going to explain exam important. Exam, NEET exam important. So here I am telling you can comment the option 20th question. Okay, just I am giving the explanation. Hemoglobin with oxygen binding is called as a oxyhemoglobin. That's good. This is a definitely reversible process. Correct na? This one is this arrow indicates the formation of the oxyhemoglobin. This one is a dissociation of the oxyhemoglobin. Okay. What are the factors favors this oxygen hemoglobin? Here, high PO2, low PCO2, low H plus ions, low body temperature. Good ma. Whoever commenting is very good. One time you check. Direct exam question only I am discussing. Okay. Once the NEET exam is over, evening we will discuss na same. We will make high PO2, low PCO2, low H plus ions, low body temperature. These are existing at the alveoli. These are existing at the alveoli. Okay. Na? So these factors at alveoli favors formation except high PO2 only one high. Remaining all are low. Next one is a dissociation, low PO2. High PCO2, high H plus ion concentration, low acidic, okay. The next one is a high body temperature. One time you can check. These are existing at the tissues. These are existing at the tissues. So, these are high PCO2, what it called low PO2, high PCO2, high H plus ions, high body temperature. These are the factors existing at the tissues favors the dissociation of hemoglobin from the oxygen. E question damn sure. I said na hypothalamus how much important, this is also important. Now I am going. Next one is this direct question, okay. What did you call 21 question? Good. Sri Bharati, third option, that's very good. Okay, Radha, third option, good, good, good. Move on. The next one is the following, 21st question, move on. The following reaction is a direct question. CO2 plus H2O, that's definitely important. Only one question. Direct answer I am telling is a carbonic anhydrase. Direct option I am telling is a carbonic anhydrase. Okay. This reaction is mediated, reversible reaction is mediated by the carbonic anhydrase. Okay. Hexokinase already I said, pyruvate dehydrogenase. Anybody pyruvate dehydrogenase reaction? Pyruvate dehydrogenase reaction, good. Option 3 carbonic, it's very important. That we can speak. Anybody? Reaction mediated by pyruvate dehydrogenase. Pyruvate dehydrogenase, Kirtana, Radha, Aisha, Balusri, Vijayanu. Reaction mediated by pyruvate dehydrogenase first. Type along with that citrate synthase. These two. Anyone? Answer, okay. Link reaction, good. Sri Bharati is a first link reaction. Tell link reaction. What is the link reaction? To tell. Pyruvate dehydrogenase followed by citrate synthase. No problem. Type fastly. Pyruvate dehydro. I Anbalgan. No, wrong. Alcohol fermentation, there is no relation. Sri Bharati, TCA cycle. Reaction you tell. This I am telling. Pyruvate dehydrogenase is converting the pyruvate to acetyl CoA. Why you are getting pyruvic acid converted into acetyl CoA? This reaction is mediated by the pyruvate dehydrogenase. This reaction, pyruvate, each pyruvate containing a 3 carbons, acetyl CoA containing a 2 carbon. There is a release of carbon dioxide and also there is a release of NADH plus H plus. This is the first oxidative decarboxylation. I am telling, this is a, so almost this we have very short time duration. You should be very active in this one. Okay, pyruvate dehydrogenase. Okay, this is a first oxidative. This is occurring in the mitochondrial matrix. This is occurring in the mitochondrial matrix. Okay, citrate synthase. Anyone is a citrate synthase? Citrate synthase, good. Kamalini Palani. Citrate synthase. Citrate synthase is a converting the, what is called as a oxaloacetate along with the acetyl CoA, condensation of the water, acetyl CoA, condensation of the water to forms a citrate. One time you can check. This pen is going very slowly. So citrate synthase, commonly that's good. Citrate, oxaloacetic acid to citric acid. Okay, na? Acrolinate. From where you got Radha? Radha, yes, acrolinate. Okay. Oxaloacetate is associated with acetyl CoA condensed with the water to form the citrate. This reaction is mediated by citrate synthase. Now I am going to give one offer to you. So, hexokinase you know, glucose 2, glucose 6 phosphate, pyruvate dehydrogenase you know, citrate. Now I am asking one question, last question with this. I am asking with this one, if you tell definitely one gift. I don't know what gift I cannot tell. So, this will be the highly secret. Okay. Whoever tell the first one. How many I am writing? Okay. How many molecules of CO2 H2O can convert into H2CO3 by 1 carbonic anhydrase per second? First question. How many molecules of carbon dioxide and water can convert into bicarbonic acid by one enzyme, 1 carbonic anhydrase per second? First commentary person will get the gift. You need to that information also, okay? Anyone. If you comes to the corporate office Tirunelveli, gift is ready. Question you understand now? Wow, Venusri. Anyone, biomolecules lo, enzymes lo, introduction. Okay? 6 lakh. What is this? Aishwarya. Kirtana is a 600. What? One time you can check the biomolecules enzyme. 6 lakh. 6 lakh. 6 lakh. Venusri. Good. Venusri, your gift is ready at corporate office. Okay, Tirunelveli. Okay, our main center, KNA main center. So, very good. Congrats. I am concluding. One carbonic anhydride. This is whatever we are studying, there is a linking with other chapter. One carbonic anhydrase enzyme can convert 6 lakh carbon dioxide and water into carbonic acid 1 second. 1 second. That is the one. So, congrats. Next one is exchange of the gases. This is a similar question. Anybody comment? Exchange of the gases at alveoli is mainly based on this question. 22nd question. There is no gift for this one. I will announce the name who commented the first one. 22nd. Some are coming and going. Be constant. So within this is the 22nd. 10 more questions. 10 more questions. Aishwarya, all of the above, good. Baulisri, all of the above, good. Move on fastly. Fast. Kirtana first. Radha 4. No problem. Type whatever you feel. Radha, RS, amalgam 1. So 4, sorry. 10 more questions, okay? 10 more questions. Okay. Venusri 4, C. Same question I made, if you remember. All of the above. This exchange of, one time you can check the breathing and exchange of gases. Exchange of the gases at mainly based on pressure or concentration gradient. Option 1. Option 1. No, option 4 is a wrong answer. Exchange of, you one time you can check and you can that paragraph. Exchange of the gases at alveoli is mainly based on the pressure or concentration gradient, full stop. Solubility of the gases, thickness of diffusion membrane are also affect exchange of the gases, are also the main factor is based on the pressure or concentration. No, it's not option 4. So, you didn't read the NCERT properly. Yes, you didn't read properly. Take the NCERT, you can check. Mainly based on the pressure or concentration gradient. After that only statement will start. So, almost everyone except Kirtana, everybody said wrong answer. Everybody said 4th option. See, a small thing. Okay, going. Okay, one time you can check NCERT. Next one. This is a conceptual, okay? Na? So, example, but breathing and exchange of gases based on this one only, okay? So, respiratory gases directly, I am only telling the answer. Without this chapter, without this table, breathing and exchange of gases explanation is incomplete. That is why, what did you call partial pressure of oxygen at the alveoli is a 104. I am going for the option 1, okay? First option, important, conceptually important, okay? Na? If we want to understand, we can. Second, partial pressure of carbon dioxide is very easy to remember. Atmosphere 0.3, remaining 40, 45, 40, 45. Carbon dioxide easy to remember, oxygen one time you can check. Okay. Next one, good. Ball is good. Venusri, Sri Bharati, Aisha, Kirtana. Okay. So, Kirtana is active. One more is what it called? That's good. Now, this question. One more question I am telling. First, hypothalamus I said. Na? Next one is the factors affecting the exchange of, factors affecting the binding of oxygen with hemo. This is a question. Example, definitely question will come. No doubt. Respiratory gases, definitely question will come. Okay. So here I am going to make the one is that directly answer you can tell 24th question, but one time you can check on to the one. What it called as a tidal volume. Next one is inspiratory reserve volume, expiratory reserve volume. Definitely question will come. Expiratory reserve volume. Next one is residual volume, inspiratory capacity. Okay, inspiratory capacity. One time check the question, Kirtana. Inspiratory capacity. Next one is a expiratory capacity, functional residual capacity. Functional residual capacity. Next one is a vital capacity. Next one is a total lung capacity. These are the volumes. Okay. Total capacity. That's okay. Answer is good. Answer is good. Answer is good. 4. Can you come to the present? There's a small gap. Sri Bharati 3. FRC is equal to RV. Correct only, na? Sri Bharati. Ball is Sri. This one, E question lo, if you did mistake, I will not accept. Definitely question will come in these respiratory volumes and respiratory capacities. No doubt about it. So total 3 to 4 questions we can expect in the breathing and exchange of gases. One question on the respiratory volumes, one question on the transport of the oxygen, one question in the introduction. Okay. So here, tidal volume, how much tidal volume? Vc is equal to, okay, tidal volume is equal to 500 ml. IRV is equal to 2500 to 3000 ml. ERV is equal to 1000 to 1100 ml. RV is equal to, sorry, RV is equal to, pen is this, sorry, 1100 to 1200. IC is equal to TV plus IRV. EC is equal to TV plus ERV. Okay, na? FRC is equal to RV plus ERV. RV plus ERV. Vital capacity is equal to TV plus ERV plus IRV. Total lung capacity is equal to TV plus ERV plus IRV plus RV. IRV plus RV. One time you can check. Definitions also, okay. That question will come, no doubt about it. Okay, na? Okay. Either this volumes with match the following, otherwise directly they will ask as a statement. More possibility is the match the following. Okay. One time you can check. TV is equal to 500 ml, IRV 2500 to 3000 ml, ERV 1000 to 1100, RV 1100 to 1200, IC is equal to TV plus IRV, EC is equal to TV plus ERV, FRC is equal to RV plus ERV, VC is equal to TV plus ERV plus IRV, total lung capacity is a total TV plus ERV plus IRV plus RV. Otherwise, VC plus RV. No doubt, question will come. We will discuss on the March 4th evening. Okay. Now, next question. The following, this already given question, this is the one. I said one question will give in the introduction, this question. They given already. I think post-COVID, just post-COVID. What are the functions of the conducting part? Okay, na? Last very few questions, be active, okay, na? Or 10 minutes it will complete. First, conducting part is extending from the external nose up to the terminal bronchiole. Up to the terminal bronchiole. Okay. What are the functions? 4. This comes under the conducting part. 4 functions. One is called as a transport good. Who is somebody? Yes. Option 3. First commented by Aishwarya. Good. Aishwarya is. Who is this Aisha madam? You are telling for the 25th question only you are telling that. 4th option you made, one time you can check. Don't delete. Just you can check. Okay. Good. So, transport of air from atmosphere to the alveoli, one function. Extraordinary functions. Next level you will study. The next one is clears foreign particles. Humidifies. Next one brings the air is equals to body temperature. I wrote fastly but important. 4 function. Taste, that is they are not speaking. Okay, na? The smell, sorry. Taste or smell, sorry. So how can you taste? No. So we can smell. That is why 4 functions, okay? Transport of the air from atmosphere to the alveoli. Clears the foreign particle. Dust particles you can see. Bacteria also we can remove. Humidifies, mix with the air, mix with the water. Moist. Making the inhale air temperature is equals to body temperature. These are the 4 functions of the conducting part which is expanding from the external nose up to the terminal bronchiole. So, I am going for option 3. Site of diffusion. Site of diffusion primarily by the alveoli. Not only alveoli, alveolar ducts. Okay, going. Next one is, so I am going forward, okay. Next one is, okay. Tarun Sujit, sir, Malayathilo, Vannam, Parayamu, please at least one line. Enda Vena Mone. Okay, Tarun Sujit, Enda Vena Mone. Okay, Poduma Malayalam. Okay, then we are going to the 26th question. Okay, 26th question. Among the following, this is for you. Now, this if you tell first name, I will announce. Third option, function exchange part. Okay, that's very good. That question already went, sir. 26th, you can concentrate. This is a question for you. Among the following statement about the glomerular filtration is incorrect. Anyone? Thank you, sir. Tarun Suji. Oh, thank you, Tarun. Anyone? 26th, incorrect. Incorrect. First one is given by the Sri Bharati. That's nice. Any other? Anyone? A small thing I changed, you know. First urine formation lo, there are the 3 steps are good. Aishwarya also placed. First option said by the Sri Bharati. Okay, nice, congrats. Okay, other persons are inactive. Okay, that's anyhow. Urine formation lo good. Urine formation lo, there are 3 steps: glomerular filtration, tubular reabsorption, tubular secretion. First step is a glomerular filtration, correct. Next one it is, so why it is considered as a ultrafiltration? All components of plasma except proteins undergo filtration. All components of the plasma except proteins undergo filtration. That is why it is considered as a ultrafiltration. GFR is equal to 125 ml per minute or 180 liters per day. Correct. Second statement, you know, here I said each kidney, na? It's not the each kidney, it is a kidneys. Kidneys, both kidneys. You understand the difference? 1100 to 1200 ml of the blood is filtered by the kidneys per minute, which is equals to the 1/5 of the cardiac output. That is why. Okay. So, I hope you are okay. What is the change? This is for you only I made. Because some persons will make line to line, they will read for that one. Okay. Next one. We are going to complete. This is a good question. Till now they didn't give in the exam, but 27th question. Conditional reabsorption of the sodium and water. Conditional reabsorption. Direct answer, anyone? 27th. Conditional reabsorption of the sodium and water. Conditional reabsorption. Aishwarya, good. There's a small gap between me and you. So, no problem. I will wait for everyone. Sri Bharti, good. That is one more. Very good. Actually, it's very highly conceptual. Directly, I am telling DCT. That's good. That's good. That's good. So, anybody remaining? DCT. So, directly, I am telling 2 or 3 chapters definitely it cover. DCT. When ADH, here one point I am going to clear. One point I am going to clear. What it called as a mandatory reabsorption of water and sodium. Mandatory. The word is not given, but we should remember. Because they mention conditional means mandatory, we have to remember. Mandatory reabsorption of the water and sodium is taking place at the PCT. Conditional reabsorption takes place at the DCT. Okay. At DCT, when the ADH binds, then only water reabsorption. When there is a ADH, then only DCT involves the water reabsorption. when there is a aldosterone. Ashok sir, aldosterone. Oh, AA. Okay. When aldosterone is present only, DCT involves the reabsorption of the sodium. Where if there is no ADH, DCT not involved in the reabsorption of water. When there is no aldosterone, DCT is like silent. Just like some persons will be stay in the online, but they will not comment. They will not tell the answer. Just they will observe. Silence. So when there is a ADH, when there is a aldosterone, then only DCT involve the reabsorption of water and sodium. That is why this is a directly I am going for the conditional reabsorption. Where is the mandatory reabsorption of water and sodium? PCT. Okay. Next one I am going forward. Reabsorption is a minimum, whether you are framing or not, one time you can check. Reabsorption is a minimum at ascending limb of Henle's loop. Direct, one line statement, Henle's loop lo first statement. Tubular reabsorption lo. Reabsorption is a minimum in the ascending limb of Henle's loop, but ascending limb of Henle's loop very important in the counter current mechanism. Okay. So 28th I am going for option 2. Any doubt is there, you can check, comment. Okay. 27th, okay, I am going to the 28th question, okay. Next one is a 20, this question also important, highly conceptual, this is what it called as a counter current mechanism, cardiac cycle, erythroblastosis, fetalis, counter current mechanism, these are very highly conceptual. In the mechanism of concentration of the filtrate, following play significant role. Directly I am giving the option 3. Henle's loop and vasa recta. Question number 29th I am going. 29th I am going. Okay. In the mechanism of concentration of filtrate, which of the following play the significant role? Say, but top question. Functionally top question. Henle's loop and vasa recta. Henle's loop and vasa recta. So 29. Just I am framing. Just can you wait? Can you fastly somebody type what are the short way you can make comparative difference between the cortical nephrons, JG nephrons. This is the time for you. Cortical nephrons ki JG nephrons ki differences plus among these two. Okay, if you cannot type among these two nephron, which nephron mainly involved in the concentration of urine? At least this one. At least this one. Which among the cortical nephrons and the JG nephrons, which nephrons are mainly involved in the concentration of the urine? Your turn. Aishwarya, cortical nephrons, vasa recta absent in the cortical nephrons. Aishwarya, you made two options. Both you said cortical nephrons and juxta. Can you confirm with the third message? Cortical or juxtamedullary? Balusri, Venusri, all are Sri only. Confirmation answer is a juxtamedullary nephrons, because in juxtamedullary nephrons, Henle's loop is a long, which enter into the deep layers of the Henle's loop, what it called medulla 1, and it is a well developed Henle's loop. That is why I said Henle's loop long along with the vasa recta also developed. That is why JG nephron, juxtamedullary nephrons are play very important role in the concentration of the urine. That is why I said Henle's loop and vasa recta play important role in the mechanism of concentration of the filtrate, are very important in the counter current mechanism. Okay. So next one is that. Just this question is the last one. Cortical nephrons are highly reduced. One time you can check, ma. Okay, na? Extend in the largest cell. Good, ma. Question is 29th. We are going to the close to the one. How many times? Last question regarding this one in this slide. Urine is concentrated than initial filtrate. Initial filtrate. Your turn. How many times humans urine is a more concentrated than initial filtrate? Initial filtrate. Your time. Good. First answer, Aishwarya. Venusri, 99%. What you said answer, you understand, Venusri? 99% of the filtrate are reabsorbed. Question is, how many times urine is a concentrated than initial filtrate? That is a 4 times. 4 times concentrated. 4 times. Extraordinary one. Initial filtrate is a 300 milliosmolarity per liter. Our urine is a 1200 milliosmolarity per liter. 4 times. 3 4s are 12. 1200. Mahan, no brother, 180 liter to that's I'm not asking about that. I'm asking about the urine. So here somebody said 4 times. Aishwarya, congrats, ma. Okay, na? Okay, going forward. Very superficial questions only. Even I didn't ask any small logic questions also. Next one. 30th question. Get ready. Option 1 or option 2. Which of the following enzyme convert angiotensin in the blood to angiotensin 1? Angiotensinogen to angiotensin 1. Option A or option B? Pronunciation, both are same. Option 1 or option 2? Everybody come fast. Only 3 questions only remaining. Kamalni. Option you can type, ma. Aisha madam just came. Hi. Aishwarya. Venusri. Okay, good. Anyone fast? Small thing. This only thing is that RENNIN. This pronunciation is a renin only. This is a milk coagulating enzyme. Milk coagulating enzyme present in the stomach of infants. Stomach of infants. Renin, RENNIN. Spelling both pronunciation same. RENNIN is a milk coagulating enzyme present in the stomach of the intestine of the fetus. RENIN, this is a converting the angiotensinogen to angiotensin 1. Angiotensin. This mechanism, very important, okay. So 30th, nicely, almost everyone made it correct. So good. So next one is, which of the following statement about a human? This is a last question almost. We are very close to the end. This is a 31 question is a for you only. Remaining question just I will tell you and we can go. 31st question you can check. Which of the following statement about mechanism of urine formation in human nephron is correct? Correct. 31. Yatham sir. Okay, that's good. So 31st question. Anyone? 31st. Glomerular filtration is an active process. This you can see in that last content. Okay. ANS is a very good. Okay. Descending lymph is which one we want? Correct. Correct. Yes. Very good, ma. Very good, ma. Very good. This such a question we can accept. Excretion law, there is a one possibility of the based on the conceptual. Good. Glomerular filtration is not active process. Glomerular filtration is a passive process. Glomerular filtration occur without energy. Without energy. It is a passive process. There is no energy is invested in the glomerular filtration during the urine formation. Just glomerular pressure only it will take the one. This one, this is also, I don't know how much you will understand. It is there in the content. Last is there in the contest. They will give that one. Okay. So, hi sir, Sukumano, I am your KNA student, Vineet, Suresh Manula, hi brother. Okay. So here this one, glomerular filtration is a non-selective process. How many members will not understand? I don't know. It is a non-selective process. Okay. In PCT, reabsorption of the glucose is the NCRT direct. This one is active. Glucose, amino acids, sodium reabsorption takes place by the active. Option is a 4. Descending lymph is always confusing only. Descending lymph of Henle's loop is a permeability to the water and almost impermeable to the electrolytes. Ascending lymph is impermeable to the water, permeability to the electrolytes. Always confusing statement only. But extraordinary statement Henle's loop. E statement. Because this point only determine the counter current mechanism. That is why, okay. Here no explanation. The volume of the blood pumped by each ventricle during the cardiac cycle. Is it stroke volume or cardiac volume? Directly I am going for the stroke volume. So, stroke volume is defined as a volume of the blood pumped by each ventricle per 0.8 second or per cardiac cycle. Cardiac output is the volume of the blood pumped by each ventricle per minute. Per minute. That is a stroke volume is a per cardiac cycle. This is a per minute. So, cardiac output is equal to stroke volume into number of beats per minute. So, stroke volume, that is a cardiac output approximately is a 5 liters to a common person. Okay. That is why 32, last question we are going to make. Okay. During inhalation, pressure inside the lungs is lesser than the atmosphere, because it's a direct question I am doing. Okay. Last statement I am going to tell. During inhalation, the pressure, inhalation and exhalation, that is the process of breathing, inhalation and exhalation, what it called is based on creating the pressure gradient between the atmosphere and lungs. During inhalation process, the pressure within the lungs is lesser than the atmosphere. During exhalation, the pressure within the lungs is greater than the atmosphere. How much pressure difference we will study. Okay. So, going for the 33 is a 3. So, next one. This is the most important. Okay. So, I am going to conclude with a simple statement. Those who are taking the repeaters. So, whoever, those who are preparing for the NEET exam, single one. So, thing is like till last day, don't lose hope. Fight until the last day, last minute. Okay. 1. The last one is this word. The one which convert average into the excellence is a consistency. Whoever consistency. The rarest character of the human is a consistency. That's right. Okay. So, thank you very much for supporting everyone. Okay. So, thank you.